Hello students, my name is Akash. I am IISER alumnus, and today we are going to discuss one of the question that has been asked in IAT 2023. Let's see this question. It's from the chapter biomolecule. So this question actually covers the concept of enzyme and its inhibitors, basically competitive inhibitor. Let's see the question. So they are asking that the diagram represents an enzyme, its substrate and potential inhibitors. So P, Q, R and S as well as T are the inhibitors of this enzyme. Okay. Now they are asking which one of the following combination is the best pair of competitive inhibitors for the enzymes. So they are asking which among these 5 inhibitors comes under the category of competitive inhibitors, or which may come under the category of competitive inhibitors. Okay. So in order to solve this question, we need to first know what are competitive inhibitors. So let's see. Competitive inhibitors are basically those molecules which actually resembles the substrate, whose shape is very similar to that of substrate, and they actually compete with the substrate to bind with the active site of the enzyme. Okay, so let's say this is a substrate. The inhibitor should also look like this, which resembles the substrate. Okay, and now both of them will try to bind on the active site here, and one of them will of course bind to this active site. Okay, so now the catch is that out of these 5 molecules, only those molecules can be the competitive inhibitors which have the shape similar to the substrate, which is triangular in shape. So all among these 5, all the molecules which have triangular shape or triangular sub shape can be categorized under the category of competitive inhibitors. So we can see here that P has a triangular shape here. Okay, although it has some other shape as a whole, but yes, they have a triangular part in it. Now this one also have 2 triangular parts in it. Other ones are squares and circles. That's why Q, R and T can't be categorized, or they don't have potential to be categorized under competitive inhibitors, while P and S can be categorized under the category of competitive inhibitors. Okay, so the answer should be P and S, which is A option. So this is the answer. So this was the question here, which was quite simple, but it needs the knowledge of competitive inhibitors. One of the questions that has been asked in the paper of IAT 2023. So let's see the question. This question actually covers the knowledge of Hardy-Weinberg principle as well as genetics. Okay, so Hardy-Weinberg principle actually is related to genetics as well as the chapter of evolution. Let's see what is it. So Hardy-Weinberg principle says that if a population is not evolving, or if we can say that if the gene pool of a population is constant, then only it follows the Hardy-Weinberg equilibrium, which is stated here, and which is like this: P square plus 2PQ plus Q square is equal to 1. And there is one more equation, P plus Q is equal to 1, where P denotes the dominant allele, frequency of the dominant allele, Q denotes frequency of recessive allele, P square denotes frequency of dominant genotype, Q square denotes frequency of recessive genotype, and 2PQ denotes frequency of heterozygous genotypes. Now this question is saying that in a population with 10,000 individuals, 4 of them have sickle cell anemia, which is a recessive autosomal disease. Now in this question they are asking about how many of them will be heterozygous for this disease. So basically they are trying to ask about this 2PQ, and they have given us the information about this Q square. They are saying that 4 out of 10,000 individuals are sickle cell anemic, which means Q square is equal to 4 upon 10,000, which comes out to be 4 into 10 to the power minus 4. Now we can of course get the value of Q. Just take a square root of this thing, and you will get here 2 into 10 to the power minus 2, which is 0.02. Now, since you have got the value of Q, you can of course calculate the value of P, because P plus Q is equal to 1. Now, P is equal to 1 minus Q, which is 1 minus 0.02, and it comes out to be 0.98. So now we have both the things, P as well as Q. So now as we have got the value of Q and P, we can of course get the value of 2 PQ, which will come out to be 2 into 0.02 into 0.98. There will be some value here, okay. But they are not asking about the frequency. This value is about frequency. 2 PQ is the frequency of heterozygotes, but they are asking about the number of individuals. So you need to multiply this whole value with 10,000 also. That will come out to be 392. So that's why 392 is the answer here. Okay. So this was an interesting question from the topic of Hardy-Weinberg principle. Hope you enjoyed it. So this question is basically match the following type of question, and it is asking basically that which of the following parts of enzyme matches with which of things that is given in column number 2. Okay. So hem actually it's a prosthetic group. And what's a prosthetic group? Prosthetic group is a part of enzyme which is tightly bound to enzyme, and it is an organic compound. So it's something which is organic in nature and it is tightly bound to enzyme is known as prosthetic group. So this hem is actually a prosthetic group which is present in peroxidase and catalase enzymes. So that's why A matches with 4. Now NAD, molecules like NAD, FAD, FMN, all of these comes under the category of coenzymes. So coenzymes are also organic molecules just like prosthetic groups, but they are loosely bound to enzymes. There is just one difference here, and that is between the tightly bound and the loosely bound thing. Okay. Now next thing, zinc. Zinc, as you all know, Zn2+, it's a metal ion, and metal ion comes under the category of cofactor. Okay. So zinc is actually a cofactor which is a metal ion which is present in many of the enzymes like carbonic anhydrase, carboxypeptidase, alcohol dehydrogenase, and there are many more. Now next thing, niacin, it's the other name of an enzyme known as vitamin B3. Deficiency of B3 causes pellagra. So this niacin is actually a part of a complex enzyme known as B3. So that's why it matches with 2, option number 2. Hence, the correct option should be A to 4, B to 3, C to 1, and D to 2. 4, 3, 2, 1. So the correct option should be 4, 3, 1, 2, which is B option. Okay. So this was the question. This question is also from the year 2022. Okay, and this is from the topic of respiration. Okay. Now they are simply asking about the electron transport chain. You must have read about electron transport chain, how electrons are transported in the inner membrane of mitochondria, how they generate a proton gradient, and how that proton gradient is broken down to produce ATP. Okay. So they are just asking only that thing. Okay. Here in this diagram on the right side, they are trying to ask about the protons flowing across the membrane, and in this side they are trying to ask about the ATP synthesis, on which side ATP is synthesized. Okay. So we know that protons are always transported from the mitochondrial matrix to the inner membrane of mitochondria. Okay, and hence proton gradient is generated. Now this proton gradient is broken down to produce ATP, and only one option is correct here, that is A. Okay, because here in this diagram ATP synthesis is occurring on the intermembrane side, which is not correct. In this diagram this flow is on the opposite side. In this diagram again this flow is on the opposite side. So the only correct option remains the A option. Okay. This question was asked in year 2022 and this is from the topic of genetics. Now what this question is asking, let's discuss. They are saying that color of seeds is exclusively determined by two alleles. First one is capital W, which gives white color, and the second one is small w, which gives brown color. Okay. Now they are saying that brown color is recessive. Brown color is recessively inherited, while white color is dominant. Okay. So we know that the color which is dominant will be expressed mostly because only one of the alleles can give this characteristic in the progenies. Okay. Now they are saying that homozygous brown seeds and homozygous white seeds are being crossed. It means capital W is crossed with small w, and now they are asking what will be the seed color and the genotype of the embryo in the resultant seed seeds obtained from this cross. So just make a cross here. Here in the progeny side we will have one W which is capital and one small w. The resultant progenies will be capital W, small w. So all the progeny will have the genotype capital W, small w, and we know that among capital W, small w, capital W is dominant. Okay, and capital W codes for white color. So all the progenies will have white color. Okay, so all the progenies will have white color with a genotype capital W, small w. That is the answer, which is option number B. Okay, thank you for watching. Let's meet in the next video.